Good afternoon. Let's go ahead and finish up the lecture that we started today. We talked about secondary structure, and you might have noticed that we talked about alpha helices, but we didn't talk about beta sheets. And that is a big omission. You need to know about beta sheets as well as beta turns and loops, but those are much simpler to implement. You need to know less about them. You need to know less about beta sheets than alpha helices too, but first I want you to take a piece of paper and make like a 1-inch strip of it, like this. In fact, I'm going to go ahead and do this twice. What you have from this is you have what's essentially what looks like something that Jane Richardson drew. Remember the arrow with the point on one end? You know, so if this is an amino acid chain, you can imagine that it's got an N-terminus, if I can write it correctly, an N-terminus and a C-terminus. Okay? And I'll do another N-terminus and another C-terminus on mine. You can feel free to do this as much as you want to make your own protein model out of a strip of paper. When we had the helix, we had repeating hydrogen bonds that held it together. Beta sheets are multiple beta strands, so each of these would be a strand of an amino acid. Multiple beta strands can actually fit together to line up hydrogen bonds to make a beta sheet. Technically, one is a strand, two or more is a sheet. And as we'll see, they can go together with the N's and the C's in the same direction. Then the arrows would be pointing in the same way. They would be parallel. Or you can flip one over, it would be anti-parallel. And the only question is, how do the hydrogen bonds line up better? It turns out that they line up okay both ways. There's one way they line up slightly better. But I want you to have your little beta strand model with you. So this is the second conformation. Of course, they predict it by Pauling and Corey because they noticed that if you had two strands going straight, that you could start to line up the hydrogen bonds. And so actually, if you look at it, it goes straight if you look at it from the N to C terminus. It's basically going straight. It zigzags up and down. So if you turn it this way, and I'll have a picture of it, but if you turn it a certain way, you'll actually see a slight zigzag up and down. It's hard to, I guess we could fold our pieces of paper to make it look like that, but that's too much big of a deal. We'll just look at the pictures. There are two kinds of strands, can be parallel or they can be anti-parallel. And just remember, a strand by itself is not really a secondary structure because it does not have repeating hydrogen bonds. Secondary structure that is held together by repeating hydrogen bonds only happens when you have two or more strands in a sheet. This is what an anti-parallel looks like. And just notice that the side chains for the alpha helix, remember that you have the curl of the backbone and the side chains are going out from it. It looked like a flower, like a foxglove flower, if you know what those look like. Beta strands, on the other hand, will be like the straight direction of the chain. The side chains will be going up and down from this. And you can see that that's how we're shown in this. You have one up and then one down, one up and one down. And they are 7 angstroms apart in the sort of default conformation. The most stable kind of beta sheet that you can get is where the strands run in opposite directions. And so we have N versus C, and here we have, I'll flip it around, C versus N. So they'll be going in directions like this, but also C, if you rotate them, then you would have the side chains going up and down. This is a three-dimensional thing. This is why I want you to actually have a piece of paper in front of you. And I don't know if you can come up with some way to even, like, this would be the side chain going up, you know, and then you'd have another one going down, then another one going up. 7 angstroms between the two pointing in the same direction on the same strand. So just notice that that makes them anti-parallel, and the hydrogen bonds line up really well this way. You can see that they are straight on, plus, minus, plus, like we were talking about. Or actually, it's the plus is in the middle, so it's minus, plus, minus. Okay. You can make it work also if they're parallel, and it turns out this changes the dimensions slightly. The hydrogen bonds, as you can see, are kind of crooked, and so they're not as strong. If this happens, it's not as much as of a stabilization force, but other stabilization forces can fit into it. You also have the up down of the strand, but the strands are shifted slightly relative to each other. They go up and down a little more. And so because of that, the side chains are brought slightly closer together. They're 6.5 angstroms. Notice that the strands are all going in the same direction. One important thing, look at these strands and be able to confirm what the direction of the strand is just from the atoms. Look how you have N, C alpha, CO, N, C alpha, CO. And that tells you the direction of the strand. Each amino acid has an N-terminal end and the C-terminal end, and those correspond exactly to this N connected to the C alpha and the CO connected to the C alpha. It all makes sense, but I've noticed that students on tests, sometimes I'll give you something like this. I'll take away the arrows and I'll say, is this parallel or anti-parallel? There'll be some students that can't do that. So be ready to do that, because it's actually not that bad. You just look for the NCC and you move in that direction. You know the N is the N terminus. I'm spending too much time on this, but I want you to get it right, okay? The thing about the strands, if you look at them from the side, when you have multiple strands together, they start to get a little twist. In fact, you can see it very clearly in this model of E. coli thioredoxin. And this twist is actually, it can go either direction. It can be left-handed or right-handed. It's not predictable from the peptide bond, but I think some people might have predicted it just in general, that there would be a twist, not its direction is predictable, but the fact that there would be a twist. Because if you just think about the physics of a strand, I want you to take your piece of paper and you see how you can hold it to the side, and I want you to feel how easy it is to bend that piece of paper. If you have a flat piece of paper, easy to bend. Now I want you to twist it like this. You see how I'm doing that? And then I want you to sort of compress it, and of course you're still strong enough to do this, you know, but you can actually feel that it resists you a little bit now. It's stronger because it's twisted. And so if you have a bunch of strands that are twisted in the same way, they can line up just fine, and they can gain more strength for that particular beta sheet. Now, if you just looked at this picture, you'd be able to tell that you actually can have left-handed or right-handed twists, because you have both of them represented in this figure. You can twist in either direction, and so if you look at this figure, what is the handedness of the overall twist? Now, you have to establish an overall direction, but 4 of the 5 strands are going in the same direction. So you can establish that and move your hand along it, the direction that's established, the 4 of 5, so it's up is the direction that you move it in, and then you curve it. Now, I'm not sure if I am the same way as you. I might have my camera flipped, okay? But if you do this, you will see, I believe that it is a right-handed twist. For all of the strands, for the whole sheet as a whole, because 4 of the 5 strands are right-handed, but there's one strand that is actually left-handed. Is that right? Because you're moving in the other direction, and you're twisting in the opposite direction. That might or might not be true. I'm not going to ask about the twistedness of a single strand. I'm not going to be that picky. But I want you to see that overall this is definitely a right-handed twist for at least the 4 parallel sheets. And I've already answered the question of, is this parallel or anti-parallel? Well, it's both. It has a parallel side and an anti-parallel side. The side with the strand 4 in it is anti-parallel to the other ones, and so it is going to be an anti-parallel. Mostly, well, it's an anti-parallel for that half, and then it's parallel for the other half. So it wouldn't be fair for me to force you to have an answer for that. So alpha and beta are the main things that Richardson had to draw. And so most proteins have a lot of alpha or a lot of beta, or they have both. And so you can have, you can actually add it up because there are particular psi angles that go with the particular kind of secondary structure. And so you can actually run a computer and you can say, does it fall into the psi angles that are alpha or beta of the secondary structure? And you can define it, and you can also just look at the protein and you can sort of look at it and confirm the computer's right. So you look at this one on the right and you say, you see both a beta sheet and a parallel alpha helix structure. It's a parallel beta sheet and a parallel group of alpha helices. I believe these are called, this is placental ribonuclease inhibitor. It's an interesting protein. I think there are some proteins that are infinitely repeating proteins, potentially, that have just a repeated alpha-beta sequence. But you can see this is one way to get a parallel beta sheet. So over time, you can actually have a sequence that can even change from alpha to beta, especially if the conditions change. Because if it's sort of on the knife edge of whether it's going to curl up or go straight, then you're going to have you could have the conditions change, and it could tilt the balance where it turns from alpha helix to beta sheet. In fact, we know this can happen because we can look at old paintings. Old paintings have been covered with an egg whitewash. That's a really good way to have a clear layer, and it turns out that dries out and makes a really sturdy kind of layer like varnish. It's natural varnish. Over time, so you start with ovalbumin protein, and it's an alpha helical mostly protein. So you paint that on the painting, and it's mostly alpha helical in that clear egg layer. Now, over time, actually, if you look at old paintings, they don't have as much alpha helix content. They've decreased by 73%, and beta sheet content has increased by 44%. In other words, those strands, as they dry out and sort of harden over time, actually turn more into beta strands, and those combine into beta sheets. And it turns out this is what makes egg whites be such a good painting varnish, because the beta sheet is more closed off to oxygen. Oxygen has a harder time getting through all these sheets. Basically, it turns from a curl like this to a wall like this that has all of these connections, and it's like a fence that literally keeps the oxygen out. You can do detection of what is the secondary structure in that protein layer, and you can see it for yourself. So there's two other kinds. Remember, there were two other colors in the model. I have it around here somewhere. Here it is. But you've seen that before. We talked about the orange alpha helix. We talked about the blue beta sheet. And now there's green, which is a beta turn. Can you see that? This is a very short turn. It's like making a U-turn when you're doing that, like at the intersection by the bridge. If you have to do a U-turn, you know that you have to turn your wheel really tight. If a protein wants to do a U-turn, it has to turn its wheel really tight, and it's helpful to have particular residues that can adopt those particular angles. It turns out that that is it's a single strong hydrogen bond that holds it together, but it counts as secondary structure because it is a backbone hydrogen bond, and it is a regular feature. It just is not as repeating. It's repeating in different beta turns, so I'll give you that. It's basically a feature of the protein backbone. And so the beta turn here is going to, you can see how it snaps together the NH and the CO on the bottom. And you can have different types of beta turns, different types of sequences will encourage this kind of really weird phi-psi combination. You'll notice about type 2, look at that, there's a glycine. You should be able to recognize that by now, that you have the NCC, and then you have the side chain, which is just a hydrogen. Type 2 beta turns always have a glycine at that point. They also have prolines, because prolines restricted phi-psi bonds actually fit well with certain types of beta turns. So before we said glycine and proline are not found in the other two, here they are found in these. Forming that hydrogen bond is crucial for this to be a turn and not a mere loop. And this is where you also see proline in its special cis configuration, because the cis configuration of the omega bond is conducive to this kind of weird, sharp turn of the backbone. And finally, we have the white color. The white color is a miscellaneous. It doesn't fall into the other categories. It doesn't have repeating backbone hydrogen bonds, and therefore, we just say it's whatever it ends up being. It's loops. Here's a domain that has, you can see all the beta strands. You can count them up, and there's actually, it looks like there's 9 beta strands, although the 9th one might not be really in the strand as much. Yeah, So there's 8, 9, or 10 beta strands in a sort of repeated sheet. This is a sandwich between two beta sheets, in fact. You can see that. And the loops at the end, they don't have any secondary structure. These are too big to be turns. They are not the same thing as turns. They don't have any defining hydrogen bond structure. And they're not necessarily flexible, but they can be. Okay? It looks like that loop is just waving around. And in some cases it might be. But it also could be a well-defined structure where those side chains are holding it in place and it's just not getting any hydrogen bonding from the backbone. You don't know until you do it. The one thing about loops is because they're at the ends of the protein and because they don't have to form particular structures, they are highly variable in both sequence and structure. So if you think about it, remember the sequence comparisons that we would talk about. We talked about how sometimes you can put in gaps to make the sequence line up better. The gaps are going to be in places that have more variable sequences, and more variable sequences also have more variable structures. They are often loops. And so you could actually see this in a sequence comparison. You might have a homework problem that has to do with that. So after all that is done, you can look at this. We said that there are particular phi-psi angles that fit with particular secondary structures. This is a plot that shows you a Ramachandran plot showing which secondary structures are associated with which areas of the graph. Remember how we had 3 windows, 2 major windows, 1 minor window that was even open on the graph. Notice that you have the alpha helix is in the bottom one of these, and the beta sheet is in the northwest one of these. There's a northeast one, a northwest one, and a southwest one. Alpha helix is on the southwest quadrant, and so it's going to, when you are curling up in the alpha helix, you're going to be adopting a negative 60 phi angle, and that's psi, negative 60 psi angle, and then an angle of phi that's negative, okay? So be able to read a graph like this and notice what it says. Alpha helix has different phi-psi angles than a beta sheet does. Anti-parallel and parallel beta sheets are right next to each other. So you can see that those have very similar angles. And it doesn't really matter if they're right twisted, parallel, or anti-parallel. They're all in the same area. The other thing that's in this area is the collagen triple helix, which we will talk about in the next video. So it's really important, not all helices are alpha helixes. The collagen triple helix has phi-psi angles that look more like beta sheets. It is more of an extended structure than it is a curl. It's not a very tightly wound helix. These are all ways of saying the same thing. So the right-handed alpha helix down there is very different from the collagen triple helix, and you can tell that by looking at the phi-psi angles. Don't look at the words if you have structural numbers to look at, you know. The other thing is the left-handed alpha helix that is rare and that only happens in that little window over there. Maybe a few residues can get into it. It's a lot easier to do with a lot of glycines. So glycines do actually show up, but pretty much only in left-handed alpha helices because their phi-psi angles make that easier. And so you can take a Ramachandran plot and you can make a point for every single residue. Once you get a structure of a protein, you can do a Ramachandran plot of it. It's one of the ways that you check that the protein is right, because if the angles are wrong, you're going to have problems with that protein. It's going to be the wrong structure. You know physically that they have to be at least close to these wells. And you see here the rabbit pyruvate kinase. Most of the residues fall within those areas, and even the ones that don't are kind of close. They might be places where the protein is genuinely stressed into a weird phi-psi angle. More likely, they're mistakes in the structure. Proteins don't stress that much, usually. And if you have this many, I'm guessing that later, better structures of pyruvate kinase have more tightly, most of these ones that are outside the wells, they're probably mistakes from the structure, but they're close. Here's examples of good-looking and bad-looking Ramachandran plots so that you can know if you're looking at one. The one on the left is one that looks good. You see it's all in the allowed areas, and it's even in the favored areas mostly. There is one that comes from the suspicious one. You see how it's not really, it's a majority in the right areas, but there's a lot of things outside that area. That is most likely a mistake in the building of the protein model. And let's see, obviously bad. The one on the right is kind of bad. You have too many things that are just on the edge. So you can compare the left and the right for kind of bad on the right and definitely good on the left. Okay. And by the way, they give you the percentage of residues in the favored and outlier regions. And the lower percentage, 96.2, is lower than 97.8. And that's why you can say that the right structure is worse than the left structure. And you can see that the middle one is just ridiculous. You don't want to do that one. All right, so you have a Lecture 6 worksheet. You can't quite finish it yet. It has some concepts that will come later, but let's go ahead and give you those concepts. Remember to start reading. Read the news article. You can do that this weekend, and you can start to read the intro, the abstract, and look at the figures. Don't look at the results of C et al., our main reading. Don't look at the results section just yet. You can skim it if you want, but we will really talk about it in earnest in the lectures on Monday. And that finishes up Lecture 6. Now it's time for mini lectures.